Hello everyone, welcome to Supplement Tutorials. Today we'll be talking about the Amoeboid Protozoans. And before we begin, I'd like you to hit on the subscribe button to subscribe to this channel. Please, it's very important to subscribe to the channel. It helps us produce more videos of this kind. So please, I'm begging you, just hit on the subscribe button to subscribe to this channel. Alright, now before we start, we are going to take a quick overview of our previous lecture to usher us into this lecture. And in our previous lecture, we talked about protozoology, and we were able to discuss or analyze most of the phylums that fall under protozoans. And we're able to talk about the Sarcomastigophores, the epicomplexans, the Celiophora, etc. Today we are going to take one phylum and then take a look at a subphylum within that phylum. So the phylum that we are going to look at is the phylum Sarcomastigophora. Sarcomastigophora. And under Sarcomastigophora, we are going to look at the Sarcodines. Under Sarcomastigophora, we have Sarcodines and then Mastigophores. In other words, these are the amoebas, and then these are the flagellates. Okay, so today we are specifically going to talk about the Sarcodines or the amoebas. Now, the amoebas, as we already know, belong to the kingdom Protostista. And then they belong to the phylum, they belong to the kingdom Protostista, and they belong to the phylum Sarcomastigophora. And then the subphylum Sarcodina, as I have illustrated here. Now, these amoeboid protozoans have general characteristics that runs through them, both those that are medically important and those that are not medically important. So these are the general characteristics. We've grouped them into according to their shape, their locomotion, their feeding, etc. So the first characteristics we have to look at is their shape. So we know that they are irregular in shape. They do not have one specific shape, and it's because of how because they lack hot cell wall. So basically, the Sarcodines are eukaryotes. So they have membrane-bound structures within them. So what you have to know is that they are eukaryotes. Eukaryotes. So they have membrane-bound structures within them, and then they are irregular in shape. Now, their locomotion is using their pseudopodia or their pseudopods. So pseudopods mean the pseudo means force, and then the pods mean feet. So like force feet. So you see the amoeboid, the amoebas, they look something like this. So they have force feet. So they extend their cell membrane in order to cause them to locomote or to move. Okay, and then their feeding is the holozoic nutrition. So when we talk about holozoic nutrition, it's all about phagocytosis, or what do we say, endocytosis. Let me put it that way. Phagocytosis is more of phagocyte. Endocytosis. So what they do is that when they target a particular organism, they extend their cell membrane around that organism, and then they form an endosome around that particular organism, and then their hydrolytic enzymes within them digest that organism. So their feeding or their nutrition is mostly what? Endocytosis. And that process is what we call the holozoic nutrition. Then under reproduction, they mostly undergo, there is a short form of reproduction, which is also known as the binary fission. So normally what this organism does is that in arid conditions or in severe conditions that they may not be able to survive, they undergo what we call encystation. And this encystation allows them to survive through this harsh condition. And through the encystation, they undergo the asexual reproduction by producing, their offsprings of their own kind through cell division. And after that, they encysst to produce what we call the trophozoite. We'll get to know about it. And then the respiration and excretion, they undergo respiration and excretion through diffusion. Okay, so they take in oxygen within them through diffusion, and then they give out the wasteful product through excretion. And most of these organisms are free living, especially in freshwater bodies. When we talk about the amoeba proteus, the amoeba proteus, which is not pathogenic. Proteus, the amoeba proteus, for instance, lives in fresh water conditions, and there are some parasitic amoebas like the Entamoeba histolytica. They are mostly found in the human gastrointestinal tract. So that is the habitat for you. Alright, so now we are going to take a swift look at some of the parasitological significant amoebas of parasitological importance. Alright, so we are first going to begin with Entamoeba histolytica. So Entamoeba histolytica is a common amoeba parasite that is responsible for what we call amoebiasis, or what we call amoebic dysentery. So transmission is mostly oral fecal. So when you take in a contaminated water or a contaminated food, the infective stage is the cyst. So when you take in an infected water or a contaminated food, what you take within your body is the cyst. So it is within your gastrointestinal tract that this cyst will encysst and then form trophozoite. And the trophozoite is the actively growing and feeding stage. And the trophozoite is what is going to cause all the observable symptoms that you encounter during amoebic dysentery. So this is the life cycle. So first of all, there is cyst formation. So once the cyst has been formed, remember the cyst is mostly passed through stool. The cyst is mostly passed through stool into water bodies. So when that water gets contaminated and then you drink the water, you've taken in the cyst. Now within the gastrointestinal tract, especially the large intestines, the cyst will undergo what we call Encystation. And then Encystation will also lead to the release of the trophozoite. And still within the gastrointestinal tract, the trophozoite will also undergo Encystation, and then will form cyst. So first of all, we have Encystation. That is here Encystation. That is the formation of cyst. So this is what actually happened during harsh conditions or arid condition. The trophozoites undergo what we call Encystation. So they round up and then they form what we call a cyst. And during Encystation, that is where the binary fission occurs. So when they encysst, what they do is that they divide themselves into 4 nuclei. So you realize that they will divide themselves into 4 nuclei like this. And then each nucleus will form a membrane around itself. And then during the Excystation, they exist, or they burst out to release 4 trophozoites. So that is about the life cycle of the Entamoeba histolytica. Now, how are we going to diagnose Entamoeba histolytica? So mostly we use stool microscopy. Now, what we have to notice about the Entamoeba histolytica is the ingestion of RBCs. So you realize that when you observe Entamoeba histolytica under the microscope from a stool sample, you realize that within its cytoplasm there are red blood cells, and that is an indication of Entamoeba histolytica. I already said that they undergo holozoic nutrition, which means they engulf pathogens through endocytosis, or they engulf substances through endocytosis. So what they also do is that they consume red blood cells. When they see red blood cells, they throw their membrane around the red blood cells, and then they undergo what we call endocytosis. They capture the red blood cell in them. So that is one diagnostic significance or diagnostic marker that you'd use to know that this particular organism within the stool is Entamoeba histolytica. The presence of RBCs in its cell membrane. Now, we have other diagnostic method that we use. We have the PCR, which is often the gold standard, and then we have the ELISA, that is enzyme-linked immunosorbent assay. Okay, so symptoms of amoebiasis, we have bloody, diarrhea, and then abdominal pain. So that is basically the Entamoeba histolytica for you. Now, we have other species of Entamoeba. We have the Entamoeba coli. The Entamoeba coli is structurally similar to Entamoeba histolytica. But as I said earlier on, the only thing you use to differentiate between Entamoeba histolytica and Entamoeba coli is the ingestion of RBCs. The moment you see RBCs within the cytoplasm of the Entamoeba histolytica, is to tell you that it is Entamoeba histolytica. Yeah. But if you don't see any RBCs within the cytoplasm, then it might have been contaminated with the stool might have been contaminated with Entamoeba coli. So Entamoeba coli is non-pathogenic, and there is a minute difference between Entamoeba coli and Entamoeba histolytica. Now let's go to the other Entamoeba. We have the Entamoeba gingivalis. So the Entamoeba gingivalis is mostly found in the oral cavity. So it's mostly found within the teeth. And the transmission is through direct contact. It could be through kissing, intense kissing, or sharing of, let's say, spoons, etc. So anything that got to do with the mouth, could transmit Entamoeba gingivalis. So they are mostly found in the oral cavity, and they are morphologically similar. They are all amoebas. So when it comes to the morphology, they are similar to that of the Entamoeba coli and that of the Entamoeba histolytica. But how are you going to know that this is Entamoeba gingivalis? The site of transmission, or where it resides, its habitat. So it's mostly found at the teeth. So if you're able to isolate sample from the teeth, and then you see this amoeboid organism under the microscope, we should tell you that it's none other than Entamoeba gingivalis. It is mostly not pathogenic, but it's associated with poor oral hygiene. So it's mostly associated with poor oral hygiene. Now our next Amoeboid protozoa is the Entamoeba species. The Entamoeba species mostly causes two diseases. So we have the Entamoeba, or someone will say Entamoeba keratitis. So the keratitis causes severe eye infection, and then we have the granulomatous amoebic encephalitis. So for this one, it's got to do with brain injury. So this organism is one of the most fatal amoebas that we have, though it's rare, but it causes this severe brain infection, mostly found in children. Okay, so that is the Entamoeba species. Then we have, of course, I would say this is the most dangerous amoeboid protozoa. So we have the Naegleria fowleri, that is also known as the brain-eating amoeba. So this brain-eating amoeba causes the primary amoebic meningoencephalitis, primary amoebic meningoencephalitis, also known as PAM. And this is a very serious brain disease. And one thing special about the Naegleria fowleri is its life cycle. So it is the only amoeboid parasite that also has a flagellated trophozoite, or a flagellated stage. So it has an Encystation stage, then it has an amoeboid stage, or amoeboid trophozoite, and then finally a flagellated trophozoite. So it looks as if it undergoes through the 3 stages. So it can somehow be classified as a mastigophora anyway, but it is mostly placed under the amoebas. So the trophozoites enter the nose and then travels through the olfactory nerve to the brain, causing severe brain damage. This thing can be so fatal, and people who are infected with this particular disease can die within weeks or days. Alright, so this is just a brief summary, or this is just a summary of the amoeboid protozoans. So the amoeboid protozoans, those that are of parasitological importance, are really not much, but then they somehow causes severe infections or severe illness, especially in immunocompromised patients. Alright, so thank you very much for joining us in today's session. I would like you to hit on the subscribe button to subscribe to this channel for us. If you enjoy this video, you can share the video and then you can like it as well. Thank you very much for enjoying this session.